Today we're going to be talking about how to inoculate tubed media. Tubed media usually contains a broth or some solid medium like agar that's nutritive for the bacteria. The first thing you need to do is, of course, label your tubes properly, and that should include the organism that's in there and any identifying information for yourself and the lab that you're in. Here we see unknown gram-negative bacillus, a date, initials, and lab B. The next thing you'll want to do is flame your inoculating loop. This sterilizes it by incineration. This is a loop. You can see it has a smooth loop at the end which holds a droplet of inoculum with living bacteria in it. When you flame your loop, you want to do it as vertically as possible and get as much of that wire glowing at the same time as you can. Here's another example of that before we do an inoculation. So next we'll pick up the tube, remove the cap, briefly flame the neck of the tube, and then insert your sterilized loop into that inoculum. You should swirl the loop around the inside of the inoculum to get a good bead going, but try not to touch the inside of the tube if you can avoid it. Here we're going to transfer inoculum from a broth and then streak it onto the surface of a solid media called a slant. It's called a slant because the solid medium is set up inside the tube in a slanted orientation, which you'll see in just a moment. So you flame the loop, put the loop into the tube, get a bead of inoculum, and then get the recipient tube. Take the cap off, flame it, and then transfer the inoculum to the recipient tube. Here you'll see a detail of how that is streaked over the surface of the slant. You want to do it in kind of an S pattern with one pass. So we'll streak the surface, flame the tube again, and recap it. It's important at the end of any procedure like this to flame the loop again because that still has living culture on it, and if you lay that on your countertop, your work surface, you could contaminate that surface. So always terminally flame your instruments. Here we're going to demonstrate the same procedure, only using a more traditional tube called a screw top tube. This one is a little bit trickier in that the top is smaller and you have to hold it with your pinky. When you're doing this procedure, you want to try as much as possible to not put caps of tubes or lids of plates directly on your work surface. That could contaminate your tube. So we try to hold it in our instrument hand and then move the tube in and out of the cap. Here you'll see a detail of how that's done. Notice that the pinky holds the cap and you screw the tube out of the cap, perform your inoculation, and then put the cap back on by screwing the tube into the cap. In this last example, we're going to demonstrate how to transfer inoculum from a flask to a tube. It's not terribly difficult. You simply remove the cover or plug from the flask, whatever the case might be. Reach into the flask, trying not to touch the inside surfaces of the flask, get your inoculum, and then transfer it to whatever recipient tube you have. And as always, terminally flame your loop.